Thank you for staying. Let's see, let's get my pointer and hopefully we're set. That way. So I'm going to be talking to you about the Texas E. coli Bacterial Source Tracking Library. This is the, hopefully, what we've been developing over the last 8 years as a statewide library. And I tend to call I didn't know quite what to call myself, so I said the curator. But really it's the girl with the cryo gloves, staring at the computer screen, crossing her eyes at the genetic fingerprints while mumbling at Excel. So that didn't quite fit in the bio sketch, though. So when I think about the Texas E. coli BST library, I really think of three aspects. One is that it is an archive, a living archive, literally thousands of frozen cultures of E. coli that have been isolated from water and unknown source samples from across Texas. It is a database. Those are the ones that cross my eyes. This is a database of over 10,000 genetic fingerprints. And more importantly, we have used these to devise a tool. Now remember, bacterial source tracking has a goal, which is to identify the sources of fecal contamination. And so the current version of the Texas E. coli BST library, through all of these archives, databases, we have screened to try to find so far we have 1,393 isolates from 1,232 different source samples that we have screened. We've tried to self-validate. Dr. Harwood was talking about promiscuous strains. I tend to call them cosmopolitan, non-judgmental. But the whole goal of this is to identify sources of fecal contamination so that we can aid in the best management practices, the wastewater protection plans, the total maximum daily loads. Now, there's been a lot of, you've heard this already, why E. coli? And there's been a lot of arguments. Maybe we should use it as a target organism, maybe not. However, we still have to remember that we have the levels of E. coli have regulatory significance, that we have established monitoring and standard methods. We actually do get the EPA gold medal seal of approval for those. And there's still sometimes an uncertain relationship between the library independent targets and those E. coli levels. So yes, this is E. coli. This is the more general slide that you've heard of today. My talk sort of goes through my personal history of source tracking that Dr. Steckel went through this morning can also be seen here. So we are looking at E. coli or bacterial source tracking. These are tools. A lot of what you've heard is most of the E. coli BST methods are library dependent at this point, where you need that database of reference bacteria from known animal and human sources. And the idea is that in order to capture the diversity of animals in a watershed, in order to capture the diversity of the E. coli in the animals, you're going to need a large local watershed that would be considered most useful in some of these to actually identify isolates. So when we were young, we tried to do that. We had two early Texas BST studies, the Lakes Waco and Belton that was sponsored through Soil Board, and then the TCEQ study of the San Antonio study, which was several watersheds. These were large. We we archived, this is where this 25,000 number comes in. We've archived, 3,000 E. coli isolates, 765 source samples. So these were large studies. But the goals of these studies, of course, first of all, why are we here? Identify contamination sources. These were the early studies, so we wanted to standardize those protocols, and we wanted to compare BST methods. So by standardizing protocols, you start at the very beginning, and that's with the source sample collection. Now, our goal was to maximize diversity. In a way, this sort of shoots yourself in the foot when it comes to library statistics, but we are scientists, it's for the cause. We use a sanitary survey, and also I think is very important is those stakeholder concerns. That first BST tool is the eyes on the ground, and those are the stakeholders. So, and also, they won't believe you if you haven't addressed their concerns. And say, yes, we did look into that. So get that input early. We collected a high number of source samples, and we didn't say, cha-ching, we found a feeding lot, let's get all our cows from this one. You have to get animals from different areas, and we limited it to 1 sample per animal. So when we say source, we're literally saying that 1 poop pile. That's it. I put the little asterisk there. Sewage was 1 dip. You know that's from a lot of people. And then we also isolated the E. coli through the water, using the water compliance methods. So even though these are E. coli from fecal sources, we use the water source methods. All right, so we archived 5 isolates, and we screened the 3. Now, again, I wanted to emphasize that we're isolating E. coli from source samples and from water samples using the same EPA gold standard methods. Sometimes when you isolate isolates from fecal samples, you tend to use clinical media. You tend to get some enrichment. You're selecting a different source of a different population source. Now, if your goal is to find what E. coli is in the water, then when you're using your known sources to develop your reference library, you need to get that same population group. So this is why it's important to use those same methods. We're using the modified MTEC and the NA-MUG. And we do have some experience with E. coli that we have adopted from other the cultures that we have adopted from other studies where they didn't use these methods, and then we've screened them using these methods, and some of them were not E. coli and some of them were MUG negative. So you have to be very careful about how you, what you incorporate into your libraries. The other goal of these studies was to determine what BST method or combination of methods were best. There's different ways to think of these methods. You can think of them in terms of working from lowest discrimination power to highest discrimination. Pulse field gel electrophoresis. Dr. McElhenney was talking about this earlier today with her food source tracking studies. They use the pulse field gel electrophoresis for the outbreaks. So you are zooming in very tightly on a specific E. coli isolate. We have less discrimination down here, and these two are in the middle. There's also costs considerations, of course, and effort considerations, which is also time is money. So when we're looking at, the other thing to think about is looking, this one study where we used all 4 methods, you can almost think of them as eyewitnesses to a crime. So you have 4 different perspectives, each from a different viewpoint. And so that as you get these descriptions in, you know who the perp is. And the more they come together, the more confidence you have in those results. So the first thing we did with all these E. coli isolates from these different source samples was to send out the clones, decloning. Again, this is a shot in the foot as far as statistics goes, but the key here is to maximize the diversity of the isolates that are in your library. So we screened using the ERIC PCR technique, or ERIC PCR method. We took the 3 isolates from the 1 fecal sample. We looked at their pattern. If they were greater than 80% similar, we considered them clones. That 80% number came from our QC sample that we run over and how, what's the word? Reproducible that pattern is. So that was running about 85% over time. It's been a long time. So when you run that QC pattern over time, and then compare those patterns, they're about 85% similar. So that's what we considered. Then they're the same pattern if we have anything 80 or 85 and above. So we kick out the clones. We use at least 1 isolate from each library, or 1 isolate from each sample. And then when we were first developing these large libraries, we tried to make it as representative as possible. So if they were already in the library, regardless of source, then we just looked at the unique ones or used the most abundant. When we looked at data matching, this is a little different than some of the other methods that you've talked about, and you've seen those clusters of groups. Our approach is different. Our approach is more the best match to a single isolate in the library. So you take the pattern, you compare it to all the rest of the patterns in the library, the one that's the most similar, that's the match. We don't talk about how they're grouping at that point. Now, although it's a match to a single isolate, we sort by host class. So we know that that isolate came from a cow, but perhaps we just look at it as livestock. Perhaps we just look at it as non-avian livestock. Perhaps we just look at it as non-human. So that's where some of these source classes come in. And then again, the best match, if the best match is only 50%, 70%, that's not a match. Those are different patterns. So again, that QC data comes in handy, or is used, came in handy. The QC data is used to identify the similarity cutoff. All right, so if it's below that similarity cutoff, we consider it unidentified. So we will get an unidentified category. Now this is for water, but we're also, when I say an unknown, I'm either talking about a water isolate, an isolate that came from a water sample that we don't know what it's from, that's what we want to ID, or, which way are we going? There we go. Or in using library accuracy tests, these jackknife tests, the rates of correct classification. This is the way of seeing where you pick 1 isolate at a time, you treat it as an unknown, in other words, it's not in the library, and then you compare it to the rest of the library and see if it comes up as a match. All right, and again, since we didn't let clones in here, the isolate can't not go back and match its sister isolate. We did do these 4-method tests. This is sort of a compare of what it looked like, and with applied bionumerics, you can actually do a composite test, or a composite data set. So this is the way of bringing all those eyewitnesses together. And these are unweighted, so we give them all the same credence. All right, so not surprisingly, the 4-method composite gave the best results when we looked at laser. The 4-method composite gave the best results when we looked at the internal blind QC study, as compared to the single different methods. Again, you get those 4 different viewpoints, and you have a lot more confidence in your results, and they were able to ID well. When we looked at the jackknife or the cross-identifications using the four-method composite, if you see here, for example, this is human, so your rate of correct classifications over there at like 83%. This is cross-identifying, so anything in red is a human, and so you can look at the specificity here, and also, well, this is the sensitivity, and then you can see what actually sort of came up as human, what random would have been, and then where the human actually showed up with the other isolates. And so what you see is that for each category, it came out right. So you have high sensitivity here. The place where you see where it's a little above random is where the other livestock, avian and the livestock sort of cross together. And now, if you look at these source categories, that's not the matches, that's how we sorted them out. So one of the keys that we'll be talking about later is how do you divide your matches? Where's your confidence level? We use these source classes because this is what they wanted with the study. And we're trying to figure out how to do this. The four method worked really well, except it's four methods and it's really expensive. And so what we looked at is if we could try a two method and get the same congruence. So you have the matrix that you develop with the four, and then you have the matrix that you do with these different combinations, and what's most similar. And we saw that the ERIC RP gave very similar results as the four method, but without the expense of the four. So from this Waco-Belton study, of course, the first key is, why are we here? To identify the source contaminations. So cattle was suspected as the main source, but it ended up that it was wildlife and livestock, and then wildlife, then livestock, then human. So it was different than what we expected, but we were very sure about these results. While PFG had the highest rate of correct classification for any single method, it could only identify 20% of the water isolates. So it's very good at determining foodborne pathogens, but we're going down to the level of Aunt Bessie's cow, and that's not where we want to be when we're source tracking. Although big furry animal isn't where we want to be either. While the four method had the highest accuracy and was able to identify the water isolates, from this point forward, when we're looking at time and cost considerations, we stuck with the ERIC PCR automated riboprinting methods. Okay. So the next step is to say, all right, we've just invested in these two large studies. We've standardized our protocols. We know what methods to use. Let's see if we can develop a library and use it for the state. So the first thing we wanted to do was refine the library and take out any of those isolates that didn't work, that didn't match. So on a local library, so we did this for Waco-Belton, we did this for San Antonio, we did that jackknife analysis. If they cross-identified, we cut them out, getting rid of those cosmopolitan strains. We're using the most stringent of the 7-way splits to do that, as far as trying to match them. We did leave in the ones that were left unidentified to increase the diversity of the library. And again, we're using the 80% similarity using the ERIC RP composite data set And then when we combine those libraries, we get the self-validated library isolates. And we felt really good. We had strong data. And we're going to challenge this and see how it works. So, yay, we ended up with 1,009 isolates that were source-specific. And that we had good average rates of correct classification. Yay. So we decided to challenge this with the Lake Granberry source samples. Now, my dear friend just talked about the Lake Granberry study. So here we are with the Lake Granberry source samples. This is just the 80 isolates from the known sources, and this is what it looked like if you did it all by itself. Now we decided to treat them as unknowns, and in this case, these are extreme external isolates for the challenge. They're from a completely different library or a different watershed, completely different time they were collected. And of course, we got hug. But if you look at this using Dr. Harwood's notations here, we're about 18% benefit over random. It is above random. But we can do better. We want to do better. So we need to ponder this and expand the library. So that was version 1.0 of the library. We're now on version 8.10. We've included the self-validated isolates from 7 watersheds now, not just those 2 studies. We're a little larger, but something that I wanted to show you now. So here are the statistics. They're even prettier. Look at those. Small cross-reactivity here. Large sensitivity. Way better than random. These black bars are the random. So this was our library, Texas Ecoli library jackknife analysis. Now in here is now these Lake Granberry isolates, the 44 that were correctly identified in its own little jackknife, the 16 that were unidentified, but hey, they're diverse. You don't want to throw out the diversity. How can you identify water isolates? And so we include those in the library. So now what you're going to see is the inclusive. And again, we had to step back and say, what are we trying to do with this library? And the goal is to try to identify sources of fecal contamination in these different watersheds. So don't kick yourself in the foot. Wait, I don't know if that's even possible. Okay. But don't make it harder than it has to be. So what we're doing is we're including the self-validated local source isolates in the statewide library to represent some of those watershed quirks. The fact that there was more septic in Lake Granberry than sewage, when a lot of our samples from Waco and Belton and San Antonio were sewage for humans. So again, this is a tool. So what we find then, when we use it as inclusive, where you include the local self-validated isolates, is that you get back to the results of those that are similar to the small local library, but this number left unidentified, which you come over here, 28% left unidentified here, 11% left unidentified here. And again, the point is identify those water isolates, the isolates in the water samples. So you'll have fewer unidentified isolates, which will mean you increase the water IDs. Now, the way we do rates of correct classification is over attempts, identification attempts. So when it's left unidentified, you put it in the unidentified category, and then you look at, did it identify it correctly or did it not? And that's the percent of those attempts, that's how you get this ARCC number. All right. So again, we have sufficient confidence in this. But we're always working to try to make this work better. But as Dr. Steckel was saying, we seek out our flaws. And so this is actually our worst of these challenges. But bare my soul, here it is. But that's where we learn, and this is what we've learned from that. Oh, I went backwards. All right, so one of the key things when we're looking at these source tracking is that we have a lot of confidence in these three-way source tracking splits of human, is it livestock, is it wildlife? Very confident in that. That's really not what the stakeholders need or want. So we need to, while considering the biology of the E. coli, that there can be cross-IDs, that there's a large variety of wildlife that you have to look at, that you have those cosmopolitan strains, the biology of the E. coli, and sometimes these geographical and temporal differences. We can look at maybe doing a six-way split of humans, pets, avian livestock, non-avian livestock, wildlife avian, wildlife non-avian. We're still playing around a little bit with how to best group these. The advantage of our best match to a single isolate method is that the match is the match, that's it. But then how do you identify or classify those matches or those sources? And then again, whether you divide it into 3 or 6 also depends on the number of water isolates. Again, that's the whole point. You're trying to identify. So if you have 20 isolates and you're trying to divide it up into 6 categories, your statistics are going to be skewed. So we're going to look at Lake Granberry again, this time with a 6-way split. And when you look at this, you see the calculated rate of correct classification. First of all, look at these numbers, teensy tiny. 3 isolates, 6 isolates, they're small. But you look at the rate of correct classification, and you look at the composition of the library. If I was just picking an isolate out of the hat, am I doing better than just picking an isolate out of the hat? And the answer is yes. So, 2-fold to 3-fold difference over random. So this is a starting point. And it's, what we've learned from this is that for the Texas E. coli library, we add the limited local isolates, and that's the way to make it give us confidence. It decreases the number of unidentified isolates. It supplements those difficult to get wildlife isolates. And we actually do have rhino in our library. It came from the zoo. We have all sorts of crazy things. But he looked awfully mean, and sometimes it feels like that, I'm sure, Tony, when you're trying to get these wildlife isolates. It decreases the known source sampling. If you want to have large libraries are just too expensive and too costly. But this way, you can decrease the known source sampling and processing from hundreds or thousands to hundreds. So you're saving that time and money. We keep additionally adding more isolates every time we do a study, and we keep refining. So for the future of the Texas E. coli BST library, we're going to continue to expand. We're going to continue to do those challenges, and we're going to continue to try to refine this library. We're now on version 11.11. We have more studies of Leon Lampassis and Dr. Gentry's group. We now have expanded our infrastructure so that we're not the only one doing this. In Texas, Dr. Gentry's lab is also adopting these same methods, looking at more studies. But some of the ideas we have is to try to identify these cosmopolitan promiscuous strains, no cosmopolitan, although maybe after the talk. So perhaps we'll now have another category for human, livestock, wildlife, unidentified, cosmopolitan. We're working on it. To maybe perhaps develop probabilities for strains that are frequently but not always associated with specific sources. And then, of course, we want to explore the synergy of library-dependent and independent tools. So we maybe could use the best of both worlds here. One's not from Mars, one's not from Venus. We can actually work together. Dr. Sadowski, I know that compared to Dr. Sadowski's numbers of 1 million frag DNA tags and all this, compared to my 25,000, but I'd like to show you my supercomputer, which is all our collaborators, my lab mates, past and present, as well as our funding agencies. So it's just me and the Excel sheets. And all these people. So if you have any questions, that's me with my crossed eyes. I'll be happy to take them, and I'm sure we can get other people up here to answer your questions better than I can. So any questions about the source tracking library? Yes?
What proportion of the isolates